smFISH accurately quantifies levels and localization of specific RNAs. This method is particularly useful for analyzing cell-to-cell and allele-by-allele variation in RNA. Gene transcription is an essential process in cell biology. smFISH allows for the investigation of RNA content in response to external stimuli at the single-cell level and provides spatial information.
Following estrogen receptor agonist treatment, wash the cells with cold sterile PBS with calcium and magnesium and fix the cells with 500 microliters of fixation buffer for 30 minutes on ice. At the end of the incubation, wash the cells 2 times with cold sterile PBS with calcium and magnesium for 3 minutes before adding 500 microliters of 70% ethanol to each well. Then seal the 24-well plate with a paraffin plastic film and place the plate on a rotator at 4 degrees Celsius for at least 4 and up to 16 hours. For RNA-FISH hybridization, at the end of the incubation, wash the cells 1 time with 500 microliters of 2x SSC buffer supplemented with 10% formamide for 5 minutes at room temperature on a rotator. During the incubation, place a piece of paraffin plastic film unexposed side up into a glass petri dish cleaned with an RNA surface decontaminant and place 2 sterile water-saturated paper towels along the edges of the dish. At the end of the incubation, place evenly spaced 30 microliter droplets of freshly prepared Greb1 intron and Greb1 exon probe buffer onto the clean side of the paraffin plastic film and use sterile forceps to gently place one cover.